Sophie Lin is an academic fellow at the Melbourne School of Psychological Sciences. She uses Bayesian framework, behavioral paradigms, and neuroimaging to understand decision making. She also investigates human cerebellar electrophysiology using MEG and EEG. Working together with MEG experts nationwide, she's building a whole head wearable MEG based on optically pumped magnetometers, which is the first in Australia and actually in the Southern Hemisphere. So we're very proud of that. So in this event, she will be providing an introduction on wearable MEG and also illustrating with some applications for cognitive neuroscience.
Thanks, Mara. And it's a pleasure to be here, of course. So basically, I'll kickstart with the imaging and methodology sections by, just like Mara says, introducing wearable MEG, or as you might have already heard of it, the name OPN MEG. And also, like Mara has said, we are currently building the first Australian OPN MEG facility here in the Parkville campus. And so for today's talk, I want to share with you what kind of research capability this new technology can bring to Australian neuroscience community when it arrives. But before I start, a very brief recap about neuroimaging, which we all know that track the brain in action. And to do it non-invasively, there are basically two main different ways we can do it. The first one is making use of the properties that neural populations talk to each other and also talk to the external environment using electric currents, and we can detect that outside the skull. And basically, an MEG belongs to this category. But you can also approximate neural activity by tracking the hemodynamic change, which Jacob will talk more about it in his 70 MRI talk. And once you acquire your neuroimaging data, then you need a way to analyze, look at this quite high-dimensional complex data. And in Daniel's talk, he would tell you how you can apply the latest methodology to better understand neuroimaging data. All right, now back to magnetoencephalography, MEG. So the basic physics principle of MEG is that with electric currents comes induced magnetic field, which you probably still remember from high school physics. And that is the exact signal we are looking at. So the induced magnetic field coming from their brain activity. But the thing is, that activity is tiny. How tiny? Basically, it's billions times smaller compared to the magnetic fields coming from Earth. So that's the reason, at the moment, in the current mainstream cryogenic MEG, you have to use ultra-sensitive superconductors, which sit in super cold liquid helium. And with that technology, it gives you the, state-of-art combined temporal and spatial resolution that non-invasive neuroimaging tools can give you. But the need for liquid heliums are not without its cost. Basically, it comes with some issues. To begin with, you have to create a vacuum space to separate the sensors from the hair. And that actually means you have a smaller signals because magnetic fields are inversely proportional to the square of distance to its source. And inside this big scanner, there is a fixed helmet which can accommodate to most sides of the head, but are generally non-optimal to individual head. And that issue becomes especially problematic for people with smaller heads like children. And the third thing is, during scans, people have to keep themselves quite still, otherwise you wouldn't be able to know where the signals come from. And that really restricts the types of paradigm you can apply in your research. So that's imagine if we can get rid of the liquid helium, create a wearable system. That means you can place your sensor closer to the head, you get stronger signals. You can design your sensor layouts to best accommodate your research needs and also study population. And you have more freedom to move. And basically, that dream has become true because of OPN MEG. So far, you probably know the star of OPN MEG is OPN, optically pumped magnetometers. But you will probably be asking, so exactly what do OPN do to detect magnetic fields? So basically here is the single channel OPN sensor. You can see it's quite small. You can hold it in the hand. And inside that sensor, you have an even smaller gas cell, which contains magnetic sensitive atoms. And there's a laser light passing through those atoms. In that conditions, basically, those atoms would be spinning in a direction that's parallel to the laser line. And now imagine there is a small magnetic field coming from the brain. It would deviate the spin direction towards the directions of the magnetic field, neuromagnetic field, a little bit. But that state is unstable to those atoms. So those atoms will absorb part of the laser line in order to realign themselves to the direction of the laser again. And the magic is that absorption function is linear to, it's a linear function of the magnetic field. And that is how OPNs detect tiny neuromagnetic fields in a regime totally different from the cryogenic MEG. So basically, you now have an OPN MEG system, but what can you do your research a little bit differently from different neuroimaging modality? To begin with, as I said before, you get stronger signal. And in fact, based on this 2017 paper, it tells you on the surface of the head, you get about 8 times bigger signals compared to cryogenic MEG. And even when you go deeper, to about 10 centimeters beneath the surface of the head, you still get about 2 to 3 times bigger signal. And that actually means you get a much better chance to detect deep brain activity compared to cryogenic MEG, like the hippocampal activity or the cerebellar study that we did in 2019. And the reason OPN MEG is better to detect deep brain activity also comes from that you can design your sensor layout flexibly to best suit your region of interest, like the bitemporal and mouth sensor layout in hippocampal study, and also the posterior inferior sensor layout in cerebellar study. And this advantage that you can flexibly design your sensor layout is also a very important reason why OPN MEG is especially suitable for pediatric MEG. And when needed, you can also place your sensor quite densely together, allows you to perform detailed cortical mappings. And of course, you now have a system to give you more freedom to move, and that means you can apply more naturalistic paradigm. So one classic example is the detections of beta desynchronization signals in the motor cortex when people play a little bit ball game in an OPN MEG scanner. And nowadays, it is also possible you can do a little bit hockey-pocky dance while the OPN MEG system track corresponding brain activities. But like all technologies, OPNs are not without their caveats. So one thing to remember is OPNs are not very good at detecting signals at very low or very high frequency. If that's the signals you are interested in, think twice before you decide whether you want to run an OPN MEG study or not. The other thing is OPNs, like its names, operate as magnetometers, unlike cryogenic MEG generally operate as gradiometers. It all sounds very complex, where one thing to remember is they are more susceptible to interference from the environment or even from your experimental stimuli. So that's one thing you always have to think about it before and during the day when you run your study with a medical physicist or an engineer generally will be running an OPN MEG study together with you. And I'm close to the end of my talk, but before I finish, I want to show you. So here is the room we are currently building our first full-head OPN MEG at Uni Melbourne. And we are hoping it's going to be operating at the second half of 2024. So hopefully, in the very near future, I will see you in this room running your own OPN MEG study. With that, thank you for your attention.
Our next speaker is Jacob Paul. He's a research fellow also in the School of Psychological Sciences, conducting research on the behavioral and neural correlates of numerical cognition and maths learning. His work uses visual psychophysics, high-resolution 7 Tesla neuroimaging, and computational modeling to map individual differences in the representation of numbers in the brain, including in learning disorders like dyscalculia. As a senior member of the Cognitive Neuroscience Hub, Jacob will provide an overview of the cutting-edge research being done by the Hub, using 7 Tesla MRI to advance our understanding of cognitive neuroscience.
And thank you, Sophie, for the introduction. So rather than measuring the magnetic fields coming from the brain, we're going to be using magnets in a different way to measure blood flow with 7T MRI. So let's take our wonderful new Hub logo and consider the brain that it is. And MRI is really good at giving you a glimpse of how the brain is actually processing information in very short timescales. So let's zoom into our Hub brain here and consider all of the decisions that you make and every single moment of perception, a lot of neural activity. And as you can see, that neural activity is quite computationally demanding for the brain, metabolically. It's going to drive an amount of blood flow that's going to need to re-innervate these neurons. And it's that blood flow that we're actually measuring with MRI. So as this brain continues to think, I'll let it keep thinking. What we're measuring as that blood flow changes is the amount of oxygen in the blood. And we can do that with this very large, very potentially scary looking machine here. This is the 7T MRI that we have on campus. The one of only 2 that exist in Australia, the other being in Queensland. So you're in luck that we've got one on campus, just one building across from us. And this was put in 2014. And it allows, like I said, you to do this higher resolution imaging. You may be familiar with 3 Tesla, or in a clinical setting, maybe 1.5 Tesla. Here we're actually getting essentially the HD equivalent of neuroimaging. So what do I mean by the HD equivalent? It's hard to explain just in raw numbers, but we're talking about resolution that is less than a millimeter scale. So at that scale, we can actually look at an individual brain and actually identify individual structural components. What we're measuring is differences in these tissue contrasts. And again, to give you a sense of scale, again, because these numbers don't necessarily mean anything, 0.5 of a millimeter is about the width of this grey lead. So I'm going to show you here a scan of my own brain, and we're going to start at the bottom and scan our way up. As we do that, we can start to see the wonderful, again, HD resolution that you can start to make out particular structures of the brain as we sort of move our way through. And this is wonderful because it means we can do this non-invasively. We don't need to implant anything. We don't need to involve ourselves with surgeries, and we can make out particular structures quite clearly just by the eye. So here we have my spinal cord. Also, as we move further up the brain, the cerebellum, brainstem, and you can make out my eyes here, which is always a bit freaky to look at, and my nose at the front. The ventricles, which may enlarge as you get older. And we can also make use of distinctions between grey matter and white matter, which is where the heart of cognitive neuroscience is trying to identify differences in activity in this grey matter or white matter. That's a structural image, and here is the functional image. So this is my brain condensed into a small GIF while I'm looking at some visual stimuli. You can see it's sort of tweaking away those differences in blood flow. And again, like I said, we're getting a glimpse. We're getting an image taken once every 1 to 2 seconds, maybe less than that, maybe more than that, but essentially just a glimpse of how we're thinking. So it hasn't got the greatest temporal resolution. That's improving, but we're prioritizing spatial resolution here. And again, we're getting down to resolutions of about 1 millimeter, 2 millimeters here in the functional scale. The good thing about that is that that's the population of neurons that we're capturing, the response of millions of neurons. So not as precise as single unit recordings, but we get these nice little voxels out, these little cubes. And with that, we can look at activity patterns that change across these cubes and measure that in correspondence with what we showed the participant, what we asked them to do. So yeah, I'll point out that everyone who's involved in these studies, keep a look out for them up the top here. And if you see them in the audience later on or later today, come and say hi to them and ask them about their work. So I've used this to look at the responses, our human response to time and number in the brain, and look at whereabouts our responses to this occur, the maps of these. So we can see here that as we prefer durations that are closer to a second, the colour changes across the cortex in this nice predictable way. Same for our response to numerosity. In work with Marta and Josh, who'll present later, we're able to, with 7T as well, not just look at the cortex as it is at the surface, we can actually start to resolve the layers of the cortex. And this is where we can start to really make connections between circuitry and sort of cognitive neuroscience questions as they refer to feed-forward and feedback information propagation, which is actually what we want to be looking at with neuroscience. And we know that signals are fed back through these superficial and deep layers and fed forward in this mid layer. So if we can resolve these differences, shown here in these different colours, so this is the first time we've been able to do this in Melbourne, we can start to make some strong conclusions about information flow. Natalia and student Marie have been looking at areas in the brain responsive to pain and understanding ways in which you can associate arbitrary non-words here to particular painful or non-painful stimuli, and identifying areas that respond to the tactile stimulation as compared to painful stimulation. So here's some non-words that don't mean anything. You give them some painful stimulation, and all of a sudden these otherwise nonsense words start to have a painful connotation. Sarah is about to start doing a study that for the first time we'll start using people under 18 years of age. Up to this point we've been very conservative and tested adults. But the ability to then start to look at a very important stage of development, adolescence, where something like safety or threat mechanisms start to come in. So translating some of the existing work that she has in some very nice models into the high resolution 7T and being able to, for the first time, look at people under 18, very exciting. Also for anyone considering doing some more developmental work, also a chance to talk to Sarah. The MRI machine's also able to measure spectroscopy, so not just activity of bulb responses, but the concentrations of these different neurochemicals across the entire spectrum. And work done by our hub coordinator, Janet, and co-authored by Olivia, has looked at these different concentrations of excitatory and inhibitory neurochemicals in response to a very well-known psychophysics task. So again, trying to map neurochemicals to behaviour. The scanner is also capable of resolving DTI, so white matter tracks, the transformation of information and the transfer of information. So this is work done by some people down again at the 7T scanner. But if you're interested, please get in contact with them. Some very new work and quite exciting work is using real-time neurofeedback. So here you're able to scan the brain while also taking into account what the person's response is, update the display to try and change their neural response, and train this loop of feedback from behaviour to imaging, which is quite a powerful potential clinical application. And with it, we don't just have a participant lying there still, stationary in this tube. We have a screen that we're showing them information, but we can also record all the things we're very used to in cognitive neuroscience. So recording their pulse, their breathing rate, their button box responses to stimuli on screen. We give them headphones because it's quite loud, and we can also control for their visual acuity inside and outside the scanner. Now, having collected all this extensive data at this very high resolution, very high dimensional data, you're going to need to do something with all of it to make sense of it all. And thankfully, the university has a large cloud computing resource which we can make use of to do all of these analyses, which will bridge into Daniel's talk about this. And so come and say MR hi to any of these bobbling heads throughout the day, and please roast me for that in the meantime. The other thing it's useful for is being able to produce very high resolution three-dimensional scans of your own brain. So if you are interested in getting a three-dimensional scan of your own brain, please come and participate in any of our studies, and we can very easily print you one off. Thank you.
Daniel is a DECRA fellow and head of the Prediction and Decision Making Lab. He combines neuroimaging, psychophysics, and computational modelling to investigate how decisions unfold in the brain and how our visual system makes sense of the world. And Daniel will be talking about some sophisticated methods that enables us to extract really interesting information from the brain.
I'll be talking about using machine learning in the hub to investigate how the brain relates to cognition and behavior. And thank you so much to Sophie and Jacob for being two very hard acts to follow as well. As they've mentioned, we have a beautiful suite of neuroimaging methods available at our disposal. So we have the Majestic 7T and also MEG, as Sophie explained, but also EEG, so electroencephalography, where we put electrodes on the head and record electrical activity, typically generated by cortex. We also use, in some cases, more invasive methods. So we can use stereotactic EEG with epilepsy patients to look at local sort of populations of neural activity within the brain. And with this, I guess when using a lot of these techniques, we always have in the back of our mind the types of neural responses that actually drive these sort of voxel or sensor-wise measures. And essentially, if you've had the lucky opportunity, like I have, to listen to a single neuron as it responds to stimuli, it sounds a little bit like an old sort of World War II radio. It's very crackly, it sort of fires randomly, and it's not always a very reliable indicator of what is going on in the organism's internal or external environment. And this has shifted and it sparked a nice change in thinking away from the idea of grandmother cells, so one cell that signals puppies and another that signals Anthony Albanese, to a more sort of distributed population code idea. So we have sort of large ensembles of neurons, which in concert might provide some more reliable and sort of higher signal-to-noise ratio information about the world. And it's these types of distributed patterns that we typically use machine learning in the hub to investigate the brain. So here are three sort of different images of, I think this might be a 3T MRI. That's nice. But essentially here, as Jacob mentioned, we have these three-dimensional voxels that we can measure blood flow or other measures in the brain. And so with machine learning methods, rather than looking at, say, blood flow in one of these individual cubes, we might look at these distributed patterns across voxels and how they might also differ across the conditions that we're interested in. And with EEG, we can do a quite similar thing. So here's a sort of distributed pattern, or what we call a butterfly plot, in response to a face image. And here you can see across these different electrode sensors that there's this really beautiful sort of structure that emerges. And we can look at subtle differences in these distributed sort of patterns and see if this also differs across these different experimental conditions. And to do this, we turn to our old friend AI. And by this, I don't mean we just use ChatGPT and start typing things in, but rather we use sort of more tractable or well-understood machine learning methods that have concrete links with the types of brain data that we're using. And to give you a bit of an intuition for this, imagine you're trying to classify brain responses between puppies and prime ministers. And with the EEG setup, at any given electrode or channel, you might only have very subtle differences in the magnitude of responses between these categories. So here at channel 1, you can see that there's only like a very small difference, which is very hard to discriminate. And then you have the opposite sort of pattern at channel 2. And then when we look at maybe across instances in which we present different puppies or prime ministers, you can see that we might get very overlapping brain responses within a given electrode. So here on the sort of Y and the X-axis, you see that these are quite difficult to distinguish. However, when you look at the joint distributions, or these sort of almost the XY coordinates, they become much easier to distinguish based on these patterns. And this is sort of at the heart of a lot of machine learning methods that we use today, is exploiting these types of what we call multivariate patterns. So yeah, this is in two-dimensional space. As Jacob mentioned, we can get sort of thousands of these voxels or measurements at one time. And so there we need to scale it up to these, what we call a hypercube, and I've tried to sort of demonstrate that here. So we use these different measures in combination with multivariate pattern analysis to investigate some quite interesting things in the brain, and I'll go through just a few examples today. So the first, this is some work I've been involved in. Sorry, there's no bobbling heads, but I think you'll know these people anyway. And so the first thing we use this for is understanding how the brain accounts for its own visual processing delays. So as you may know, it takes time for when light hits our retina for the photochemical bleaching processes, synaptic transmission, and also sort of it takes time within brain areas for the neurons to sort of get their recurrent processing going. And this incurs various sort of levels of delay as visual information tends to move through the visual system. And this leaves our brain in a fairly tricky spot in which if there's a sort of moving image or sort of dynamically moving thing, just based on this visual information, it's actually very hard for our brains to keep up with and effectively interact with these objects as shown here. And so we wanted to investigate one of the mechanisms by which we might be able to compensate for these delays to do things like catch a ball. To do this, participants saw a screen of, they fixated on this dot and they saw these wedges which could appear in each of these locations. So the outlined ones are not actually shown in the display, but just show where the wedge might appear in the sequences. And we showed one of the things we wanted to exploit were these sort of systematic or trajectories, motion trajectories of the wedges, in which after some exposure, the participants could predict which was the next position that would appear in the sequence. We also presented these in random order, like so. And then we trained machine learning classifiers to essentially sort of detect or determine which location people were seeing based on these distributed patterns of activity in their visual cortex. So we could use this EEG data and essentially get an above chance accuracy to decode sort of what image was presented to the person. And yeah, this was sort of from fairly early on. From there, we could then apply it to these motion sequences I described. So here's one sort of described here. You can see it going around. And there was one critical condition we included. So in the head position I've shown here, this was something that could be expected by the participant, but didn't actually appear to the observer. So the final was the last one that we would present, but the observer would still would have expected this trajectory to continue because they couldn't predict when it would terminate. And we wanted to see whether the EEG patterns, when we train on these sort of random flashes, whether those location-specific patterns would more so resemble the expected but not presented location, or that sort of behind, which was presented and still going through the visual system. And so this is very hard to pass, but I'll explain it. Essentially, what we found is that the brain responses actually did resemble this position ahead of time, or even though that the stimulus wasn't presented to the observer at all. And this tended to be sort of one ahead of what was actually presented as well. So we believe that in this case, this is an example of the brain generating these sort of representations that resemble future expected input, and that might be used in some way to help account for these processing delays, especially in contexts where there are sort of predictably changing sequences of events. And this might only be specific to motion extrapolation in this case. We're looking this up. All right, so beyond these sort of very visual focused applications, we can also look at more complex things like how we integrate information to make decisions. And this is some work mainly sort of run by Stefan Borda and his collaborators. And just to shout out to Violet's talk a little bit later on as well. So for example, if you are imagining you're going past what was previously Suada on close to here, and you might be thinking, seeing an image of some pho, you might have a number of different attributes of these things, these foods, that determine whether you might purchase it or you might drop in for lunch. So this could be how tasty you think it is, how healthy you think it is, how much it might cost. And we need to integrate these different properties into some sort of representation that we use to make decisions. And so essentially, I'm not going to give anything away here, see the afternoon talks, but Violet has been looking at some sort of different how we integrate and how these are represented in the brain over time. We can also look at things more arbitrary decisions, maybe tea or coffee, or things that are driven by our preferences, and how these sort of, the encoding of these choices might also unfold over time in the brain. And finally, we can also use machine learning to get a really good look at individual differences as well. So here's an example from Marta's group in which they used fMRI responses over sort of defined networks in the brain to predict the degree of psychotic symptoms that people might show in conditions such as schizophrenia. And essentially this allowed a really nice exploratory, or not exploratory, but sort of a powerful systematic way to map the different brain networks that might be most associated with symptoms as they occur in individuals. And what I found most interesting from this is that these different symptoms tended to be predicted by activity in distinct networks. So here we see avolition, anhedonia, attention, and hallucinations. And there's some partial overlap, but also some quite sort of distinct regions that were predictive of people's symptom severity across individuals. So that's a sort of very short overview of the type of work that we're doing in the hub and the applications that we're using. These methods are being developed and sort of rapidly evolving every day, and we're trying our best to sort of keep up with that. And we're sort of really looking at some new applications to sort of explore these multidimensional spaces. And I think there's going to be some more in the student talks as well for those who are interested. And for those who want to try their hand at this sort of thing, especially with different types of EEG data, Stefan and myself have developed a toolbox that makes this a little fairly accessible and easy to do, and allows you to run these types of analyses on your own data as well. So feel free to have a play with that or ask me some questions. And yeah, I'd be really happy to chat about that as well. And yeah, thank you so much for listening.
My question is whether it's possible to combine the different modalities, for example, the capabilities of MEG and 7 Tesla.
At the moment, because OPNs, just like the other types of energy, is super sensitive to the background magnetic field. So it's still, I can say, technically not very applicable if you want to do both of them at the same time. Like are some types of different types of technology using the same physics principle, at least try to see if it is possible to perform OPN MEG outside MSR. But I think for now, if you want to perform it at the same time, because we had a discussion before that the magnetic field inside is about 1,000,000 times bigger than the magnetic field in the natural environment. So it is still something like you have to probably recall separately and then integrated the data afterwards.
Yeah, I think for MRI, the benefit would be in going across domains is that, like I said, the resolution is really great, spatial resolution, but the temporal resolution, you sort of sacrifice that for the resolution. It's a bit like taking a long exposure photography, right? The longer it's there, but the moment you move and everything else, it starts to blur. So the benefit of having an MEG or an EEG or anything else that is able to pick up a faster temporal sequence is you could have the same task or similar task, assessing the same person across these different modalities, and then throw that all into something like a machine learning and have the same person, the sort of quantified self idea across these different modalities, and maybe get some sort of individual difference out that you wouldn't otherwise get just from the single modality.
Yeah, I was lucky enough to sort of see some practice versions of this talk, and I think, Sophie, your talk got me thinking of, the hippocampus, the cerebellum. I think these are definitely the next frontiers to be mapped using these machine learning techniques, and I'm very, very excited about what we can do there as well. I think we might also be able to fuse, so I've talked about decoding methods today, which are really, really powerful in themselves, but especially with fMRI data, actually Jacob has done some work on this, where you can also use some pre-existing knowledge of how some of these responses might be sort of tuned to things like number or duration and things like that, and get quite principled and powerful models of how sort of different areas in the brain might be sort of working together as well. So I could definitely see a really good synergy of sort of all of these things. And I think the lovely thing is that some of these modeling approaches really port across the modalities. So we're lucky to sort of have experts in all of these areas, I think.
One thing we can also remember, actually, in terms of the directions of the neural activity, EEG and MEG, they are actually, basically complementary to each other. So there's one active collaboration we are thinking about actually is to compare the EEG signal from the cerebellum with the MEG signal. So it could be something that it is possible to recall simultaneously in the meantime to integrate that multimodal in order to provide more information about brain function.
Regarding the MEG equipment, is this equipment more resilient for movement artifacts compared to EEG? Like, for example, blinks or muscle movements, or is it similarly like you need to adjust them similarly?
That's a very good question. So basically, in terms of the capabilities or the shortcomings, that you will collect all the artifacts from non-neurological sources are basically the same. But the natures of the MEG means they will be few less distortions for when the signals are being conducted from different sources. That also means when you place those signals back into source spaces, you have a better capability to isolate those non-brain signal and allow you to better detect brain signal. So that has always been the superiorities in terms of MEG signal as compared to EEG. But the thing we have with respect to OPN MEG is because you actually have a bigger signal, which I didn't mention, is with big signal, you actually have a better spatial resolution, allows you to better tear apart non-brain from brain signals. And so that's the thing, and also the practical effect that you can have a better source resolution and also to tell noise from real signals in MEG, especially in OPN MEG.
Oh, thank you. And just one more question.
Yeah.
Is there any temporal limitation, like how long recordings can you do with MEG or this equipment?
So from what I know, the system nowadays are quite stable. So if the participants can tolerate that, you can actually recall the signal for. It's possible to do it a whole day, but you, of course, you have to consider that means they have to sitting or standing inside a quite encapsulated. Still, even though it's a room bigger than MRI scanner, but it's not a free environment. So generally speaking, the section is more or less going to be the same as the typical MEG scan. You will have around 30 minutes to 1 hour. But so one thing you can imagine in the case, if in the future we can go into wild and that type of location, you might be able to have a longer scan. Yeah.
I'm wondering if there's any research or any interest done into combining TMS with the wearable MEG, and if so, what are the limitations of doing kind of TMS pulse simultaneously with MEG recordings?
Yes. I haven't seen any research being done in that, but they are definitely interested because I'm one of the person who thought about to conduct that type of study. So one of the things is you will introduce magnetic interference to that. But the good thing about it is that is something you can isolate from the brain signals. So from my point of view, purely theoretic point of view, it's doable. Yeah.
Sorry to you've been the expert today. I was wondering with the room temperature MEG, I guess the electromagnetic interference seems to be a really important sort of part of this, as discussed. And I was wondering what sort of equipment might be sort of work really well with this as well. For example, if we do have a computer monitor of some kind, are we going to put this behind a lot of lead-shielded glass, or are there any sort of types of equipment that might work really well with these setups, and any that might be a little bit more tricky just due to that type of magnetic interference?
Yes, that's also a very good question. So I don't know, if people have been to the conventional MEG systems before. If you have, so basically all these electronics have to sit outside shielded room, and then you use the projectors projecting onto a non-magnetic screen. So that's also the first stage of plan we are going to do in our MEG system. And there are possibilities, or there have been some endeavors in terms of integrating virtual reality with OPN MEG. And one of the things that you have to deal with is that with the current types of goggles of virtual reality, it creates quite a big dipoles. And so it is limited. There are some other potential type of virtual inverse reality with other types of projection to be considered possible in the future. So I think one of the things we always still have to deal with is like how do we project thing in that can be inverse enough but still not create too big, unpredictable, non-brain magnetic fields. I can think about a couple of technology that, and also quite interesting, is some kind of technology that currently Monash University is developing and could be promising. And that's the details about that we can maybe discuss later. Yeah.
Thank you.
Hi, yeah, so one of the limitations of the MEG is the spatial resolution. So I'm wondering, even though you can't do them at the same time, could you and others like systematically have people doing identical tasks with identical stimuli, even lying on your back with MEG, so that you could infer based on the spatial resolution of, say, 7 Tesla fMRI, that a signal coming from MEG over a huge data set, you could start to resolve more spatial resolution. You could start to more accurately infer where exactly the signal was coming from in MEG, so that you can get better spatial resolution from MEG using that type of approach.
All right. So theoretically, actually, to answer you about the questions, in terms of spatial resolution, in theory, purely in theory, MEG should be almost limitless. So the main reason, practically speaking, currently MEG has limited is because of the distance that I talk about. Also because of the scanner, which that with a little bit movement, you will get blurring in terms of your image. So actually, even before the development of room temperature MEG, they were customized, a haircast developed in order to enhance the spatial resolution. And from what that I know, on the surface of the brain, like the motor or sensory cortex, with that type of use of headcast, you can actually get a spatial resolution about close to 1 to 3 millimeter. And I don't know, this might be a question to you, like how good is that compared to, yeah, MI?
That's great.
Yeah. And so you can also apply the same type of customized headcast for OPM MEG, totally have no problem. That's how we start with the scan in the beginning. And with that, you can also have repeated scan with no distortions of the image that would further enhance your spatial resolution. And I hope that answers your question.
Yes, subcortically, what's the current spatial resolution limit? As you go deeper, what's the current?
That's going to be always a question, like a very, very big challenge. I do agree with you. So I think in terms of once you go deeper to about the brainstem level, it is going to be quite tricky if you want to have even sub-centimeter resolution. That can be a little bit tricky. So one thing can do is the dense and sensor array that I talk about. So customize dense sensor array specifically targeting a different area based on the inverse modeling of the individual cortical mesh. But I think computationally and also economically that could be expensive. But again, it's theoretically possible.
I'll just add that, I mean, in terms of subcortical structures, we know that with hippocampus and the amygdala, it's been quite successful. So that is, I think, compared to what we thought it was possible about 10 years ago, when people were really adamant you can't see anything from the amygdala, it's been shown that, yeah, you can. It's pretty reliable.
I just have one more question, if I may. And the question is for Jacob. Interested in laminar MRI, how hard is it to delineate the different layers? And is it an automatic process, or do you have to manually mark where the delineation is?
I think Josh will talk a lot more about this. He's suffering through this hell at the moment. It is sort of quasi-automatic, but then needing manual quality control still. The benefit is that we can scan a whole volume. We can do it in 3D. We can also do just a slab through that. and then sort of trying to line it as best as possible. But this is where also, again, a large amount of deep learning algorithms has been applied to a problem which should be fairly tractable, right? Which is intensity gradients and delineating those with grey matter and white matter borders. There are a number of packages out there that all try and do the same thing. Everyone's trying to build their own mousetrap to try and do the same thing. The benefit of having pushed for this Laminar work here is, again, the sort of analysis pipelines that come with it. So, Josh has done this sort of Sisyphean effort to push it up the hill, and it means that now everyone else that comes behind will be able to benefit from this analysis pipeline. But it's not as automatic as you would like, but it is nowhere near as bad as sort of paint by numbers manual editing like you currently have to do in some other places. So.
This is a question for Dan, actually. So I know the lab is working with kind of multivariate pattern analyses and working with machine learning techniques to decode neural activity that corresponds to some behavior or visual acuity, I'm guessing. I'm wondering if you are interested, or if there is any interest in extending these techniques to more deep learning algorithms or approaches such as autoencoders, where you can kind of encode latent spaces, and from these latent spaces try and predict what the dynamics might be telling you about the visual perception or something like that.
Yeah, thank you so much. That's a fantastic question, and I think something we've been thinking about a lot. I might plug the keynote today. I think there might be some really interesting, sort of much more considered work that I might be able to give an answer here. But I guess with a lot of the machine learning work we've been doing, it's generally been this sort of weird tug of war between sort of, that sort of complexity or power and tractability as well. So some of the sort of basic versions I've shown today do use, at what is at their core, I guess, a fairly sort of simple algorithms that can find these sort of patterns in multi-dimensional spaces. And one of the reasons we like that, and we use fairly sort of what we might call linear versions of this, is because we can sort of understand, or we can map back to the neural data fairly well in order to sort of strengthen the inferences that we can draw. With deep learning models, there has been some fantastic work sort of mapping that to, for example, the structure of the visual system, which again might be featured later today. And I think in some cases, this sort of macro structure, even of these types of models, there is really interesting correspondences which go sort of between that back and forth as well. I guess maybe the challenge is because deep learning models can be quite a black box or hard to interpret exactly how they're doing something, that raises these other challenges of sort of what is the correspondence. But I think regardless of whether we can determine that or not, it's a lot of really good inspiration for understanding maybe different encoding schemes by which the visual system, for example, may transform features into these other types of spaces, and maybe opening up new types of feature spaces that we hadn't considered before, or new types of dynamics as well. So I think I just dodged your question completely, but hopefully that's interesting.
I might have a question for probably Sophie and Jacob at the same time. So there's been a couple of papers looking at layer MEG. And I was just interested to hear what, Sophie, your take is on the challenges and in doing that, and what sort of things do we need to watch out for, and also perhaps a comment on how is it really the same thing, and how reliable? Like, would you, from the fMRI point of view, would you trust that sort of works on two in
Yeah. Okay. So layer-specific MEG, I think that's the one that basically a bit like the initial motivation why people start to using customized headcast to perform repeated MEG scan, and with that data, and then applying methodology similar to the machine learning methodology, and to be able to classify signals coming from the superficial and the deep layer. So you probably imagine it is like not surprising people would try to see with stronger signals, OPN MEG can provide better abilities to discriminate different signals coming from different layers. So far, they were only theoretical work that has been done. And the result that I learned from the author is they are slightly similar, but not better than cryogenic MEG. It could be a little bit surprising. But one thing I haven't really talked about is on the single sensor level, so in terms of the noise level, noise flow and themselves, for the current incarnations of OPNs, they are still higher than the squeeze system, the cryogenic MEG. So the current explanation in terms of why we find this surprisingly and slightly disappointing result from the modeling potential use for layer-specific MEG, OPN MEG, is probably because of that. And they are, so that's the current state. But specifically, I think the signals can still benefit from enhanced signals. And how do we make a breakthrough in terms of that? That will probably depend on hardware development. And one thing is the noise flow we're currently having is not saying on the boundary that we will have, say, in 5 years' time or 10 years' time. This is a very, very rapidly developing area. And the other thing is, in terms of OPNs in itself, I'm currently, in today's talk, I only talk about one method of OPNs. There are different types of atoms, different regimes that you can use, and there are already some papers showing that they can have even lower noise level, and that might be one thing we can look at it in, say, in a couple of years in the future, if that type of sensor might be able to bring better ability to discriminate signals from different layers. And another things I can also say, maybe potentially from the software or from the computational perspective, is to look at the signals from different frequency that might be able to help us to be able to have a better ability to differentiate signals from different layers in the cortex.
So 3T, I saw a paper out this week saying you can do layer resolved 3T imaging. So that's being pushed as well. The point is, it comes back to the question, how precise do you need to be? Mm-hmm. Do you need to see all 6 layers? If you do, then you might need to go 17 and beyond, or maybe in an animal or something like that. If you are just happy to see it's deep and superficial, 3T might be enough. The MEG laminar might be enough. But I think about also the precision with which electrical signals in EEG and magnetic signals in MEG compared to the messiness of blood flow, and it's sluggish. It's always going to be in response to something else. So it in of itself, you're measuring blood volume, you're measuring the blood oxygenation. It's a messier signal just by the nature of what blood is. So I think if your questions need that level of precision, then this is the way to go. If they don't, then don't get yourself in all sorts of trouble trying to get a signal out if the question doesn't actually demand it.
Yeah. All right. So I think to wrap up, I just want to give the opportunity for all of you to say just some final words about maybe what you think the future is. I mean, we talked quite a bit already about the OPMs without the magnetic shielded room. That would be amazing. We talked a bit about the layer OPM MEG, but are there any other final thoughts on where do you see things going from now on into the future?
For me, I think we only see the start of wearable MEG. And so, like I've mentioned, there will be different types of sensors and possibly apply slightly different regime, but using similar physics. And I think that would be going to be an exciting and rapidly developing area, and to see more and more application, especially not only for neuroscientific research, but also the promise that the MEG Society try to make to clinical applications, and we might be able to see that to be truly realization in the future, maybe future couple of years or in the future decades because of the development of a wearable system.
Absolutely, and I see people from the clinical hub. I hope you're getting excited about that too. I think that's definitely something that the traditional MEG with squids didn't quite fulfill as people expected, but I really hope that with OPM will be a bit different. Jacob?
With 17, I mean, there's only 2 of them in Australia, like I said, so there isn't a big community. So I think also we're trying to get more people who are interested to think about converting either their 3T paradigms into 7T, and just greater communities. There's a budgeted line item that the school has that funds 7T. As more people start to use it, I can't imagine that there's going to be the same amount available. So I think come in now and do it before that money, potentially disappears or exhausts or something like that, or it starts to filter out. So get moving. Get moving on it is the point. Get more people using it, and with something like deep learning or neural network models and everything else, the ability to do a lot of these things automatically is increasing as well. So don't feel like it's also throwing you in the deep end. There's some knowledge here now. So I think also I'm looking forward to bringing that together and all the rest of the modalities that come along with it.
Wonderful. Daniel?
For the immediate future student talks, one more plug for them in the afternoon. I think more longer term, though, I'd probably say a lot of the future, beyond the imaging capabilities, are the people in our hub. I think this event is a really nice reminder of how broad the sort of degree of technical and theoretical expertise we have. And I think we're really lucky in that which these things are really truly complementary and click together. So if you want an idea of how some of this research might advance in the future, maybe just sort of look at pairs of bobbling heads or something and see how these, combining these might sort of lead to something greater than the sum of its parts as well. So that's probably my view, is just I'm excited to see what people do in this sort of collaborative space.